Hey guys, I wanted to record this video for you going over some of the questions from the study guide over the different parts of the cell. Um, so I have enlarged part of this, and let me just go here. I was going to do a split screen, but it's not working out too well. There we go. So you could see um, the actual study guide as well as the CK12 here. So it starts talking about, does a cell have or need a skeleton? Well, they do have some microtubules and microfilaments that helps to give them structures. And so, so it does have different types of micro-type fibers that help to give it kind of a framework for a cytoskeleton. So here I'd probably put microfibers, and it does provide some structure for the skeleton. And it provides support, it provides shape for the cell. Functions of the cytoplasm. Let me back up here. First of all, it includes everything. The gel-like material is called cytosol. So it's basically going to include cytosol, and all this, everything inside the plasma membrane is going to be the answer for what does the cytoplasm include. And it helps to suspend the organelles. It gives them like a cushion to work in. It gives the cell its shape by pushing against the plasma membrane, and it carries out many of the reactions for this function of the cell. Okay. When we're talking about the cytoskeleton, basically it helps to give the cell structure. I'm not going to go into a whole bunch of detail here. You might just want to put down it has like microfibers to give it structure. But as far as details of this here, that's not going to be on any type of test. Okay. All right, so now I'm going to go down here to the nucleus, and I'm going to enlarge that. I'm going to get rid of that one, and I'm going to pull, when this one loads, I'm going to pull it over here so we can look at it also. Okay, I'm going to enlarge this page here. So it's talking about DNA, and you all have heard of DNA. A lot of people talk about DNA with forensic testing. And DNA is basically the code that gives your cells an idea of what their function is supposed to be. It is the genetic material that makes us what we are. And both prokaryotic and eukaryotic cells have DNA. It's just that if you have eukaryotic cells, or you are a eukaryotic cell, your DNA is encapsulated in a protective membrane called the nuclear membrane. And it goes around the structure called the nucleus. Okay. So the nucleus is a membrane enclosed organelle found in eukaryotic cells. The nucleus is the largest organelle in the cell, and it contains most of the cell's genetic information. Some mitochondria have DNA, but it's mitochondrial DNA. So when I'm asking for to describe the nucleus, you need to know that it's a membrane enclosed organelle, and it's the largest organelle in the cell, and it contains most of the cell's genetic information. Okay. And then a gene, when we talk about the nucleus contains the DNA. And DNA is coiled double helixes of what's the nucleic acid called deoxyribonucleic acid. And in your genes, which are made of the DNA, in your chromosome pairs, there are parts of the DNA that code for specific traits, and those are called genes. So when it says describe a gene, it is a short segment of DNA that contains information. It's a short segment of DNA that contains information. And it does encode RNA, but it also, that RNA is used to take information out to the cytoplasm where it assembles proteins, which gives you the traits that you have. But basically, it's a short segment of DNA, and it does contain or the code for traits that you have. Okay. And so long strands of coiled up DNA and proteins are chromosomes. So the next question says, describe a chromosome. So we're talking about in the nucleus, we're talking about DNA. DNA is long chains of amino acids. DNA can be broken into smaller segments called genes, but these are very long, thin chemical compounds. And to make them easier to store, they have the ability to coil up. And when they're coiled up around certain proteins that kind of like hold them in place, they're then called, that DNA can be called chromosomes. Okay. And each chromosome, because it's made of coiled DNA, and the coiled DNA can be divided into smaller sections called genes, that each chromosome contains many genes. The function of the nucleus is to maintain the integrity of these genes and to control the activities of the cell by regulating gene expression. So basically, what happens is if there's information in the nucleus to code for a certain type of protein that the cell needs to make, the cell has a way of copying the pattern for that protein in the nucleus, and it takes the copy of the pattern outside the nuclear membrane to the cell. So basically, the nuclear membrane keeps the blueprints for whatever needs to be made in the form of genes inside the nucleus, and it protects it so it doesn't get damaged or it has any type of alterations to it. So gene expression is the process by which the information in a gene is decoded by various cell molecules to produce a functional gene product. So what that means is gene expression is the process by which the information in a gene is decoded by various cell molecules to produce a product. You can leave off the functional gene product. So basically, gene expression is where the DNA is copied in the nuclear membrane. It's taken out to the cytoplasm to a certain organelle, and then that gene is decoded and used to make different chemicals that the cell needs. Okay, now you heard me talk about the nucleus. The DNA is inside the nucleus, and when the short segments of the DNA are called genes. You heard me say that the DNA is kind of a big molecule, and it has the ability to coil up around proteins, and those coiled DNA sections are called chromosomes. But when the DNA needs to be copied, it has to uncoil So a pattern can be made off of it, and when it's uncoiled, it's called chromatin. When it's coiled up, it's called chromosomes. So this is this section that it talks about it right here. And I'll be glad to paraphrase that for you. You can put that DNA in fine strands is called chromatin, and DNA coiled up is chromosomes. DNA in fine strands is called chromatin. DNA coiled up is called chromosomes. So you've heard me mention that the cell has a covering around it called the cell membrane. Well, the nucleus has a membrane around it. Sometimes it's called a nuclear membrane. It's also called a nuclear envelope. And it is basically to protect the DNA inside its membrane. So let me move this up here. So you can have here that the nuclear membrane is a double membrane of the nucleus that encloses the genetic material. You don't have to write all, let me go back and redo that. So basically it separates the DNA from the rest of the cell and it kind of protects it. Okay. And it does have pores in it where a copy of DNA can leave the nuclear membrane and go out to the cytoplasm where the pattern can be used to make other chemicals, and that pattern leaves to the nuclear pores. Now, sometimes inside the nucleus, you'll see a small tight cluster, a thicker cluster inside. That is called the nucleolus. And the nucleolus is mainly involved in the assembly of an organelle that the cell needs to make proteins, and that is called ribosomes. So the nucleolus is located inside the nucleus, and its main function is to assemble ribosomes, which are the organelles that kind of hold the DNA pattern, the RNA, it holds the RNA pattern made from DNA in place so that proteins can be made. Now, I'm kind of limited with the video, so I'm going to let you watch some of these videos here on your own as we go forth through this. So this is not the entire cell. This is just the nucleus. The dots represent the the nuclear pores. This is the nucleolus inside here. And your DNA, either in the form of chromosomes or chromatin, will be floating in that gel-like substance. Okay. So the next structure, oh, and let me go back one on this here. There's the virtual tour of the cell. There are practice questions here. And there's just different things you can do to kind of enhance your learning about the cell. Okay, another organelle inside the cell is called the mitochondria. And it's basically the powerhouse of the cell. It provides energy for the cell. Okay. So this here is talking about ribosomes. They're represented by the dots on the rough endoplasmic reticulum. And they're basically places where the DNA is copied by another chemical called RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. So the RNA is what can make a copy of the DNA and take it out to the cell, out into the cytoplasm. And it binds to these ribosomes. And these ribosomes kind of read the RNA pattern, and it brings in amino acids, which are the subunits, and assembles them into whatever protein needs to be made. And when we go into messenger RNA translation, we're going to have a whole unit over how this happens. So we'll kind of save some of this for later on. Right now, I'm just wanting you to kind of get the basic gist of what the organelles are called and what they do. So the mitochondria are called the power plants of the cell because they are the sites of cellular respiration. And this is where they use energy from organic compounds to make ATP, which is the cell's energy source that is used for such things as movement and cell division. Some ATP is made in the cytosol of the cell, but most of it's made inside the mitochondria. So we need to highlight this here because this is where most of your answers are going to come from. Let's put that in yellow. So mitochondria are the power plants of the cell because they are the sites of cellular respiration. They use energy from organic compounds to make ATP, which is adenosine triphosphate, which is the cell's source of energy. It's kind of like its energy molecule. Some ATP is made in the cytosol of the cell. Most of it's made inside the mitochondria. The number of mitochondria in a cell depends on the cell's energy needs. Okay. For example, active human muscle cells may have thousands of mitochondria, while less active red blood cells do not have hardly any. And remember that in the pictures, the mitochondria is kind of shaped like an oval or a bean, and then it'll have these folds in here. These folds are called cristae, and that increases the surface area inside the mitochondria so more chemical reactions can occur. And that's included on your study guide also. Okay. So back up here, ribosomes are the sites of protein synthesis. Ribosomes are made of ribonucleic acid. Let's see. They have it on here. Ribonucleic acid. Let's see. Okay, they're made of ribosomal protein and ribosomal RNA. And ribosomes may be attached to the endoplasmic reticulum. Ribosomes are RNA molecules that catalyze chemical reactions such as translation. And translation is the process of ordering amino acids to make a protein. So right now, you just need to know that the ribosomes are the sites of protein synthesis, and they may be attached to the endoplasmic reticulum. Okay. And some are attached to the nuclear envelope, but what you really need to focus on is the rough ER at this point. So now that's going to take us into talking about the endoplasmic reticulum. I'm going to get rid of some of these so we don't need all those opened up. Okay. Okay, endoplasmic reticulum. There's a rough ER and there's a smooth ER, and the difference is that the rough ER has ribosomes attached, and they're always usually represented by little bitty dots, and the smooth ER does not have ribosomes attached. Um, the function of the endoplasmic reticulum is for transporting. It can transport molecules throughout the cell. It's kind of like a little subway system. And the second function is synthesis. It allows the ribosomes to be attached to it, and it helps make proteins. And lipids are also produced in the ER. Okay. Two types of endoplasmic reticulum I've already mentioned, rough ER, smooth ER, and the difference is rough has the ribosomes. Two functions of the endoplasmic reticulum. It creates transport vesicles, and it helps to move protein to their proper destinations. Functions of the smooth ER. They may synthesize lipids, which are like fats, metabolize carbohydrates, used to store calcium ions, steroid metabolism, and drug detoxification. So there's several different things that smooth ER can do. Whereas the rough ER is mainly a place where ribosomes are attached to make proteins or chemicals, and they can be formed into transport vesicles, or it helps the Golgi apparatus to move proteins to their proper destinations. And smooth ER helps to synthesize lipids or fats, metabolize carbohydrates. Okay. Golgi apparatus is basically the packaging center for the cells. And it does look like a stack of balloons or a stack of sacks. Could be called a Golgi body or Golgi complex. And it's made of 5 to 8 layers of membranes. And basically what you need to know is that the Golgi apparatus modifies, sorts, and packages different substances for secretion out of the cell. Which is this structure or this function right here. Golgi apparatus modifies, sorts, and packages different substances for secretion out of the cell or for use within the cell. And that's basically what I need you to know for right now for Golgi apparatus. This is going to go into a little bit more detail than what we have time for. Then the next structures are called vesicles or vacuoles. And vesicles and vacuoles are transport containers. They help to move substances around inside the cell. So vesicles can fuse with the cell membranes and push substances out of the cell. Sometimes they are formed by the membrane folding over on top of itself to take in liquids or solid type foods. We saw that. Types of vesicles. There are transport vesicles, which basically move molecules between the inside of the cell and the outside. Lysosomes, they break down and can digest things in the cells. So you need to know that lysosomes break down harmful cell products, waste materials, and cellular debris, and they force them out of the cells. The peroxisomes are vesicles that use oxygen to break down toxic substances in the cell. Secretory vesicles, they help to move things to be secreted out of the cell, sometimes like waste or hormones. And I'm really not going over a lot of this. I mean, you can read through this, but I need you to just have a basic understanding of what these structures do. And then vacuoles are membrane-bound organelles that can have secretory, excretory, and also may be used for storage functions in the cell. And they're different from vesicles in that vacuoles are much larger than vesicles. And this here introduces you to the contractile vacuole that's in certain protists, or the large vacuole, central vacuole of a plant cell, which we've not gone over plant cells yet. Okay. All right, so I'm going to stop the video here for right now, because that kind of goes over the main parts of the cells that you need to know. I'm not recording.
Did I fart?
I have no idea.